Welcome to 119 New Road, Avon, Connecticut, the home of thousands upon thousands of various species. You are embarking on a journey, one that will introduce you to some of these species, mainly the interesting ones. This journey, called an ecological tour, due to its relation to living organisms and their physical surroundings, will bring you to multiple places around this residential area. Many organisms, from bears to bobcats to butterflies to bats, call this area home, as do people like you and me. These creatures live together, making use of meaningful interactions called symbiosis that allow them to avoid competition and survive, sometimes even managing to thrive. Today, we will learn about the nature of science regarding ecosystems in this area, exploring topics such as evolution, genetics, molecular biology, population ecology, trophic structures, nutrient cycling, and the effects that humans have on their surrounding environment. Whether it be in a babbling brook or the wide, flowing Farmington River, hundreds and thousands of different living organisms can be found here. So, now that a proper introduction has been given, let us begin our journey.
Starting with a cool experiment. Because the ecological world is so full of interesting questions and even more interesting answers, it seems fit that at some point on this tour we should stop, think, come up with a suitable question about a species on our tour, and follow this question with a multiple trial experiment that will give us an answer. In order to do this, we need to follow a few steps known as the scientific method. The first step of the scientific method involves thinking of a biological question which the following experiment will help to answer. This is the question that we will be working with. If given the choice, are ants more attracted to sweet or salty? This is an acceptable question due to the large number of ants that can be found in 119 New Road. The second step of the scientific method is the step during which one does background research to have a better understanding of what to expect from the question. I did a little background research of my own, and came up with multiple studies that pointed towards ants, specifically inland ants, preferring salty over sweet. The third and very important step of the scientific method is to create a hypothesis that states your expectation of the results of the following experiment. The hypothesis is usually written in an if, then, because format, stating your expectations and explaining why you have them. The hypothesis is the start of the experiment. It should contain the independent and dependent variables, as well as the constants that will be maintained during the testing. My hypothesis is as follows. If given the choice between the relatively sweet molasses cookie and the relatively salty goldfish cracker, then ants, specifically the species Monomoria minimum, or little black ant, will gravitate towards the goldfish cracker, because they are landlocked ants in inland Connecticut, far away from salt water, so their attraction to the absent salty substances will be greater than their attraction to sweet substances. The independent variable in this experiment is the type of food, while the dependent variable is the ant's attraction to the foods. The constants in this experiment include the food's equal distance from the same closest ant hill, the time each food is exposed to the ant, and the type of ant that the food is exposed to. The fourth step of the scientific method is the experiment itself. My experiment takes place within 60 minutes or an hour. I checked both food sources every 10 minutes in order to get a sense of how many ants were attracted to them and how fast they sensed them. The results are surprising and a little nasty. The fifth step of the scientific method is the analysis of the results in the conclusion. Because there are more than 40 ants around or covering the cookie, while there are only at the most 3 ants around the cracker, the conclusion of this experiment is that ants are more attracted to sweet substances than salty. Because it is important to conduct an experiment with multiple trials, I repeated the same experiment in a different location, achieving similar, albeit less dramatic, results. The sixth and final step of the scientific method is the report. This is the step in which one decides whether or not his or her hypothesis was supported or refuted. Clearly my hypothesis was refuted, as the ants were far more attracted to the molasses cookie than the goldfish crackers. This experiment is one that can help in reality, for it acts as a warning to anyone who decides to leave out sweet foods without expecting ants to arrive. Pollution by humans is a very important problem in 119 New Road and the areas around it. Metal soda and beer cans can be found in and on the banks of the Farmington River, as well as in brooks and streams. Plastic waste is also often littering the ground, and sometimes there are rusty pieces of metal from old cars and such as well. Miscellaneous pieces of trash can be found, such as old tires and candy wrappers. These items are major pollutants to the water and the rest of the ecosystem, and the effects from them are profound. Fish and other animals can get cut by the sharp cans and the rusty pieces of metal, developing infections that greatly harm them. Animals can also get caught in objects such as the plastic six-pack containers and the shred object here. These objects also contaminate the otherwise natural areas, introducing foreign bacteria into the water. Pollution is and will always be bad and can easily be stopped by simply throwing away trash in the proper places.
The species Ursus americanus, better known as the American black bear, is a beautiful creature that possesses the helpful ability to inhabit a wide range of different habitats. While this bear is native to North America, the ancestors from which it evolved lived in various places around the world, making for a rich evolutionary history. The first true bear, as it is informally known by many, belonged to the genus species Ursavis elemensis. This bear was also known as the dawn bear. The dawn bear was small in size and, while carnivorous, not at all like the dominant, hulking modern-day black bear. It lived around 20 million years ago in Europe, until about 10 million years ago, when evolution finished its job and the dawn bear evolved into the species Protursus simpsoni. One reason for this evolutionary pressure to change was due to the competition the dawn bear faced with the genus species Ursavis depereti. The resulting species, Protursus simpsoni, is considered the precursor to the genus Ursus. This species then led to yet another species of bear, one that lived primarily in Central Europe from around 5.3 to 1.8 million years ago. This species, Ursus minimus, which is now, needless to say, extinct, was better known as the Auvergne bear, and was relatively small compared to modern-day bears. However, during its existence, the Auvergne bear grew in size thanks to evolution, a change that allowed the Auvergne bear to become a dominant creature in its ecosystem. The Auvergne bear was also the first of the Ursinae family, and, in turn, the first of the Ursus line. The species Ursus abstruscus stemmed from the Auvergne bear, as they were close relatives of one another. This species was a delicate, small bear, but it served as the extremely important bridge for bears from Europe and Asia to North America. Fossils of this bear have been found in the states of Washington, Idaho, and Nevada, the areas in which the first generations of the American black bear were created. This makes Ursus abstruscus the immediate ancestor of Ursus americanus. This is a great example of evolution, as the multiple generations of different bear species led to one another, starting off from a small, fox-like bear, and finishing with a monstrous, muscular bear that dominates its ecosystem, proving that the evolutionary history of this interesting species is full of generations of bears spanning millions of years and multiple continents.
One special adaptation of the American black bear is its ability to coexist with humans by eating scraps of human food and living off of decaying and decomposing foods from areas such as campsites and garbage bags, as a black bear is doing in this picture. This adaptation ensures a constant food source for the black bear, for it is aware that many garbage bags are kept outside and easily accessible, as evidenced by the fact that on many occasions the households around one 19-year-old will report a knocked over garbage can or a torn trash bag. This is an adaptation that the black bear has evolved over the many years that it has coexisted with humans, and while it may cause irritation among the latter, and many a bungee corded garbage can, it is beneficial to the bear. One of the most obvious observable traits of the American black bear is the very trait from which it received its name, the black coat. This coat is a perfect example of a mode of inheritance known as dominance. The gene that is responsible for coat color in the American black bear is called the melanocortin 1 receptor, known as the MC1R gene. This gene has two alleles, uppercase B and lowercase b. When uppercase B is present in the genetic structure of the black bear, the coat color has a greater chance of becoming black. However, the mode of inheritance that has the final say in the color of the coat of an American black bear is known as polygenic inheritance, which is a method of trait inheritance that is characterized by the interactions of multiple genes that influence one phenotypic expression. The genes that are involved in deciding the pigment of the American black bear are the MC1R gene and the microphthalmia-associated transcription factor, known as the MITF gene. The MC1R gene is responsible for coat color in mammals, such as the black bear, while the MITF gene is responsible for modifying the MC1R gene to produce two different variations of pigment. A black bear can either be dark with a black coat or light with a pale white coat. So, the black bear has multiple genotypic and phenotypic expressions that determine the trait of a black coat, while some bears in the species Ursus americanus possess white coats, one of the other variations that polygenic inheritance provides. Lythrum salicaria, otherwise known as purple loosestrife, is an invasive plant species that is invading areas in and around 119 New Road. Purple loosestrife is native to Europe, Asia, and Africa, and that is beginning to move into North America with great speed as well, thanks to humans. Because the seeds from this plant are spread easily through insects and weather such as wind, purple loosestrife is easily able to invade swamp and river areas without competition. This flowering plant comes with a great many problems, as it is detrimental to many water-dwelling organisms. First of all, the plant takes up room that other, more helpful plants need to grow, forcing many native plants to die. Because it grows tall and has dark flowers, it shadows surrounding, low-lying plants and disrupts their photosynthesis. By killing these plants, the purple loosestrife harms the organisms that rely on these plants for food and shelter. These organisms include frogs, such as this little guy. They also include waterfowl such as geese and ducks. Considering the fact that this foreign plant was brought to North America by humans and has taken hold of areas outside of its natural habitat, the purple loosestrife is the perfect example of an invasive species. Of the many endangered species that can be found in and around 119 New Road, the species Tyto alba, or barn owl, is a very interesting creature with a rich history. The barn owl lives primarily in open areas such as farmlands, hence the name, but it can also be found in wetland areas such as swamps and meadows, locations which are found extensively in 119 New Road and the areas surrounding it. The barn owl has a relatively short lifespan of only 3 to 4 years, and feeds off of a diet of small creatures such as frogs, mice, and voles. The barn owl became endangered with the disappearance of open areas in order to create towns, shopping centers, and even cities. Very few open areas that are suitable for their existence are still in existence themselves, so the barn owl has been forced to live elsewhere, a travesty that has, unfortunately, led to the decline in their population. The impacts of this decrease in barn owl numbers involve increased numbers in mice, voles, and the other creatures who lost a predator once the barn owl became endangered. These animals were better able to adapt to new habitats, a skill that would have changed the barn owl's outlook. A practical skill that every ecotourist should learn is that of population estimation. Using the density and dispersion method, this exercise is actually rather easy. You start by estimating or measuring the size of the area under question. Then you choose what species you want to estimate population for. Then you choose an even number to cordon off by a square, and you count how many of the species is present inside of the sample area, repeating this step multiple times in different locations to ensure accurate results by using averages. You then divide the overall area by the sample size. and multiply the resulting number by the average number of species inside of the sample size. Voila, you have your population estimation. Needless to say, the species that I estimated population for was Kentucky bluegrass, a type of common grass in North America. Overall, I ended up with an estimation of 4,559,520 blades of this grass inside of 618,240 square inches, the size of my entire yard. As you can see, it is necessary to include averages by sampling a variety of locations inside the area, 19 to be precise. Many factors can affect the population size of a species, whether they be biotic, living, or abiotic, non-living. The biotic factors include other species such as earthworms, as earthworms affect the quality of soil, which plants such as Kentucky bluegrass need to grow and survive. Other biotic factors include other, taller plants that surround a species such as Kentucky bluegrass and cloud it in shadow, preventing the species from fully taking advantage of photosynthesis. Some abiotic factors that influence the population size of Kentucky bluegrass include weather. Constant cloud cover can kill the plants, greatly affecting population size. Periods of drought can do the same, but on the other hand, too much rain can drown the plants and kill them as well. Another abiotic factor is the presence of certain minerals and nutrients that the plants need to survive. With too few of these, the plants can die, while with just enough, the plants can thrive. In order for the population to reach its peak, all of these factors need to be perfectly balanced. Producers are the very first organism of any trophic structure, or food web. They harvest energy from the sun and, using photosynthesis, create food that allows them to survive. Producers include vegetation such as the algae that covers the sides and bottoms of these rocks, as well as trees such as these oaks. The next step in a trophic structure is the consumers, of which many can be found in 119 Road. Consumers can be primary, secondary, tertiary, or even quaternary. Consumers consume the producers, hence the name. However, they can also eat other consumers, as the bobcat eats the rabbit and the trout eats the dragonfly. This makes the bobcat and the trout secondary consumers. One tertiary consumer, the bald eagle, eats the trout. These consumers include animals such as rabbits, bobcats, bats,
Scores 15, 13, 6. Oh!
I'm getting bats on bats on bats.
Alright, I'm just gonna go. Bats on, bats on.
robins, as represented by this robin egg, wild turkeys, eastern newts, butterflies, bees, beetles, bald eagles, rainbow trout.
Now get me holding it. You can include me in your bio video. What kind of bat?
Dragonflies, and perch. The final organisms of the trophic structure are the decomposers, those organisms that break down decaying matter and release the nutrients back into the earth for the producers to use, starting the whole food web once again, so that it is a cycle. These decomposers include bacteria, fungi such as mushrooms, earthworms,
Oh, here's a worm!
woodlice, and slugs. Some of the organisms and trophic structures maintain relationships called symbiosis with one another. These relationships can either be examples of parasitism, commensalism, or mutualism. That last one, mutualism, is one that I would like to share with you today. There exists between the monarch butterfly and the milkweed plant a sort of mutualism, where each species benefits from the relationship. The adult monarch butterfly delivers its larva to the milkweed plant, where the caterpillars make their home and eat off of the milkweed plant. The milkweed is poisonous to other organisms except for the monarch butterfly, so the milkweed provides a sort of chemical protection to the caterpillars, for predators recognize that the caterpillar tastes bad and is mildly dangerous. The milkweed, on the other hand, benefits from the monarch butterfly because each adult butterfly pollinates it when it deposits its larva on it. A recent biological study from early 2013 targeted the species Odocoileus virginianus, or white-tailed deer, and its ability to spread the seeds of an invasive plant species called Lonicera maccae, otherwise known as Amur honeysuckle. Amur honeysuckle is a banned plant species in Connecticut, as it wreaks havoc on the ecosystems in which it is introduced. The team of researchers searched the droppings of multiple white-tailed deer, discovering that the deer ingest the fruit that Amur honeysuckle bears, depositing intact and fertile seeds when the time is right. This is an important discovery, for it helps in the fight to stop the long-distance spread of Amur honeysuckle, saving ecosystems from this invasive plant species. Nutrient cycling is a constant occurrence in all ecosystems throughout the world. A great example of a nutrient cycle is right here at 119 New Road. First of all, there are all different sorts of nutrients that organisms need in order to survive. These nutrients are needed by organic living material, but are mostly supplied by inorganic decaying material. The first nutrient that all plants need for photosynthesis is carbon, an element that is abundant in the atmosphere. Plants remove this element in the form of carbon dioxide from the atmosphere at the same time that humans and other animals are exhaling the same carbon dioxide back into the atmosphere, creating a perfect cycle. Meanwhile, carbon is also broken away from decaying matter by decomposers, who are also freeing other nutrients such as nitrogen, potassium, and phosphorus. Phosphorus is another major nutrient that is used extensively by plants. The phosphorus cycle is similar to the carbon cycle, although the weathering of rock also provides the soil with phosphorus. Phosphorus is also put into the dirt through the decomposition of animal tissue and excrement. Nitrogen is another major nutrient used by plants that is found in great amounts in the atmosphere. When plant matter decomposes, this nitrogen is sent back into the soil to be used by another living plant. Lastly, the nutrient that all organisms need, water, is cycled as well, starting off with precipitation from clouds in the atmosphere. When the water collects on the ground, it evaporates back into the atmosphere as water vapor, where it once again condenses to form clouds, leading back to precipitation. Water vapor is also put into the atmosphere through plant transpiration. All of these examples of nutrient cycling are constants in 119 New Road, for without them, no life on Earth would exist. Well, I hope you finished this tour with knowledge that you can pass on to others about the happenings at 119 New Road. If this was your first tour, I hope you enjoyed it, because I know I did.